Thank you, Andrea, and it's a pleasure and a privilege to have this chance to talk to you about reproductive hormones and dementia. Shortly after I began to prepare the talk, I realized that to do this with all of the reproductive hormones and dementia would be unwise, so we've written a little chapter that covers hormones that I'm not going to talk about, because the chief hormone of interest is estrogen, and so my talk will be aimed at the role of reproductive estrogen and dementia. And what I'm going to tell you is that, as you all know, dementia is unfortunately becoming very common in our old aging society. And that the term dementia has really taken on an unfortunate role, because everything that seems demented is dementia, and we don't have very good tools for seeing whether this is Alzheimer's or vascular disease, and so then we call dementia Alzheimer's. And we lose our way when we try to follow hormone contributions to this. I'm going to end up by talking about the difference between vascular dementia and Alzheimer's dementia, and in the end I'm going to tell you that I believe that the problem is that we have not been able to prove that estrogen prevents Alzheimer's dementia, but there is good evidence that estrogen prevents vascular dementia, and that the differences that one sees that have been attributed to estrogen's effect on dementia may very well be due to protection against vascular dementia. So let's get started. Dementia is common. Here you see studies from different areas of Spain and the increase in dementia with age. I'll show you some more data from this paper, but what you want to seek is clearly that dementia is generally a problem of aging people, and I'm using the term dementia, undefined Alzheimer's vascular. If we try to separate them, as they did in this retrospective study, we can do that, and we end up using clinical evaluations of both. And so, as I'm showing you here, while in certain parts of Spain, one can say, well, it appears that vascular dementia may be on the rise, et cetera, et cetera. I don't think that this is really sustainable. I think that what we have is overall dementia, we have Alzheimer's, we have vascular dementia, and these are clinical evaluations. And that's the case, so therefore we must consider the idea that these are two different diseases with a common outcome, dementia. And if we want to understand the real underpinnings, we have to understand the disease processes that are leading to this common outcome, dementia. So there's a lot of evidence that would lead one to believe that estrogen should have a role in preventing dementia, and we'll show you some of that, and even there is preclinical evidence about estrogen potentially protecting against Alzheimer's dementia. Here you can see the beginning, again, the study, a retrospective evaluation of, in this case, overall dementia on your far right, Alzheimer in the center, vascular dementia, but in this case, now there's also males and females, and you start to see some differences in the amount of dementia in one or the other. And you start to see some of the complexity of the problem. For example, the logic is that Alzheimer's is more common in females, and if you don't see it, or dementia is more common in females. If you don't see it, they might be protected by estrogen if they've taken estrogen. So we are leading ourselves into the solution. If we look at the Cache County study, which is a very widely quoted and well-known study, which was a retrospective study of the occurrence of dementia in a county in Utah, the state of Utah. This has recently been reevaluated. So this is from 2012. The original publications were from earlier in the century, and fundamentally, the outcomes, the upgrades are not so much that there's new data, but there's a better look at the hormone replacement therapy aspect. And again, I'm talking about dementia because I don't believe that these reports, and I've read the papers carefully, they are based on clinical evaluations and standards, but there's not a word about imaging, and there's not a word about autopsies. So you have to take this as dementia for the moment. That the Cache County study now has been further extended to look at people who started hormone treatment before 5 years or not. And in those women who started hormone treatment before 5 years after menopause, they see a difference in the rate of dementia. Also, as I mentioned before, the presence of vascular dementia is definitely not ruled out. So the Cache County study has now been updated and continues to show evidence that fits with a protective effect of estrogen. And so it's really necessary to look at this more closely. So what do? You do for Alzheimer's, you must do specific imaging studies or pathology at the time of operation, or more likely at the time of an autopsy. Both of these things will show the presence of plaques of amyloid beta 42, which is the precipitated form of amyloid. Everyone in this room has lots of circulating soluble amyloid, but we don't precipitate it in our tissues. And so you have to have evidence of this before you can make the definitive diagnosis of Alzheimer type or neurodegenerative dementia. And for most people, these studies are not available or haven't been done. There is, at present, no way to diagnose vascular dementia, and we'll talk about that in a little while, with the exception of postmortem specimens, which, as you can appreciate, are quite uncommon. So, again, when people talk about the rate of dementia and the kind of dementia, these are usually a consensus of experts looking at, usually not at patients, but looking at the clinical record. The WHI's WIMS trial used consensus to decide whether they had dementia, and even one or the other, and the Cache County trial the same way. Now, if you are thinking ahead, since you know that these are older people and they are prone to getting vascular disease, you might consider the possibility that, in fact, the two diseases coexist. And in fact, we know this is true. The issue of whether one or the other is being affected is further complicated by the problem of separating the actual two disease processes, and even, as some authors have pointed out, that the two disease processes may be interacting with each other. When you have a vascular occlusion, even a small one, the tissues can't respire properly, they get oxidative stress, they get Alzheimer's. So you have to be really thoughtful about this. So what are some of the proposed causes of these diseases? This is a cartoon of what might be causing Alzheimer's, and it comes down to the idea that there is inflammation in a portion of the brain, and either directly affecting neurons or fibers of passage that are coming from one part of the brain to the other may go through an area of inflammation, undergo damage, and have retrograde degeneration, and finally the cell body over there in the temporal lobe, which was sending an axon to the limbic system someplace else, is affected. So you can see that it's really a complicated picture. We don't know what causes it. We think it's inflammation. Inflammation is normal in all of us, and it's part of the homeostatic mechanism. And now I'm telling you that it happens to only a small portion of the cells in the brain. Only 1 out of 10 cells in the brain is a neuron. And now I'm telling you that it may in fact be happening at some distance from where the inflammation takes place, and it may be in concert with local vascular disease. This is complicated. As President Trump said, healthcare is complicated. So we have looked at this, and I didn't want to put our studies into this, but because there's not time, but here you see a kind of a cartoon that shows that when you have homeostasis, this is really a small inflammatory process. When you leave your office, there's a wastebasket full of junk. In the morning you come back, the wastebasket's in the same place, but it's empty. That's homeostasis. But if you come back and the wastebasket has been broken, or the wastebasket has been taken away, that's disease. And estrogen, through its role in part of causing immunocytes, particularly macrophages, to express cell surface antigens, regulates inflammation. It's generally counter to inflammation. If you have a normal level of estrogen, you get normal homeostasis. But if you have a low level of estrogen, you can expect a failure of this brake on inflammation, this holding it back, and now you're going to have broken wastebaskets laying around in your brain. To look at it a little bit more closely, here's Tom Wisniewski's view of how one of the two components, tau, which is a microtubule assembly protein and is responsible for the shape and activity of cells, and therefore it's considered to be a developmental protein and is only found in the brain. How tau plays a role in this, and I won't make you try to read through it, but in fact the issue is hyperphosphorylation of tau. Tau, the microtubule assembly protein, cannot assemble. Think of a bamboo rod with those little places where the parts are joined, cannot do that kind of joining without being hyperphosphorylated. If it is hyperphosphorylated, then the bamboo will join in all kinds of crazy ways. And so we have looked to see whether one could affect this hyperphosphorylation. So this is work done some time ago with one of my fellows, and you can see from this Western blot that giving estradiol to tau-expressing human cells in culture, one can, with okadaic acid, that's OA on there, which is an acid that causes hyperphosphorylation of tau, one can, in the face of oxalic acid, substantially lower the hyperphosphorylation of the tau. So you have OA, and next to it have OA and estrogen, and you can see there's less of this hyperphosphorylated tau. The rest of them are controlled studies. How does estrogen do this? How does estrogen work in the brain? Well, estrogen, as in many organs, is very widely represented through all of the brain. And here is a cartoon of the findings of any number of people about estradiol or estrogen receptors throughout the brain. And I would call your attention to the ones on the bottom: hippocampus, amygdala, temporal cortex, and hypothalamus. These are the areas that are the so-called limbic system, and they interact with each other in a massive way, so that if you had damage in these things, which are, for example, the hippocampus is responsible for memory, the amygdala is responsible for emotion, the temporal cortex is responsible for processing, particularly long-term memory, and for having connections to the hypothalamus, which is the autonomic nervous system center. And so you can see how the whole gamut of what we know as dementia can be played out through effects of estrogen, as well as, of course, any number of other things. And I want to now emphasize what I said before. Despite all efforts and clinical trials, and there have been lots of them, estrogen has not been shown to be specifically protective against neurodegeneration of the Alzheimer type in humans. We have the preclinical studies. I've shown you some of them. There are lots of animal studies. There are animal models where the genes for hyperphosphorylating tau or for making excess amyloid have been put into the mouse, and then estrogen protects against that. But transferring that to a human living person has not yet been accomplished. So since that's the case, we have to look for other causes of dementia. And I've told you a little bit about vascular dementia. I've told you about the studies that show that there's vascular dementia. So what about vascular dementia as playing a role, and perhaps the role that might show the difference or might betray the difference in estrogen's ability to protect against dementia, and especially in women who have not received menopausal hormone therapy? Could this represent the reason that you have higher postmenopausal dementia in women, and less if they, in fact, have taken menopausal hormone therapy? So what is vascular dementia? And I've put on the side, not for you to read, it's tiny writing, what I summarize on your right, which is a neuropathologist of note telling you what is vascular dementia. Number one, it can be found any place in the brain. He is talking about specimens, postmortem specimens. Remember, I told you, you cannot diagnose vascular dementia without specimens looking at it. There's simply no way to do it. So he said, or he tells you, that it can be anywhere in the brain. Of course, so can estrogen receptors. It is a small vessel disease, so you might miss it if you looked with the angiograms and any number of other ways. It can be accompanied by shrinkage, and we have all seen, or if you've looked at the scans of the brains of older people, we have all seen people with shrunken brains who seem to be functioning perfectly normal. And in fact, that's partly this business of only 1 out of 10 cells in the brain is a neuron. So diseases that affect non-neurons might not show you these kinds of effects. And neuropathologically, you can have a coexistence because now you have the specimen, you can look for the evidence of plaque and tangles, as well as the evidence of degenerative vascular disease. And it's atherosclerosis or atheromas at the very least. It's typical to what you've been hearing throughout the whole meeting, so it's not hard to do it when you have the specimen. So what do we know about vascular disease? So for that, we now have the privilege of being able to look back at the huge amount of studies that have gone into understanding the role of estrogen in cardioprevention. So I've to some degree summarized it here. Certainly the most important person in the field has been Tom Clarkson, who unfortunately died about a year ago. And with his monkey studies, he showed very clearly that estrogen could prevent the occurrence of atherogenesis in monkeys that were fed an atherogenic diet. And he also showed that if the atherogenesis was already established, estrogen could not remove it. So he then coined the term, the timing hypothesis, which is to say that you must get into that window when you are interfering with accumulation of plaque, not after the plaque is there. You cannot do that and get rid of it with estrogen if you want to use estrogen to do it. After a long time and lots of hard work, Dr. Hodis published recently the results of a 5-year, this is important, 5 years of treatment with estradiol and with or without progestin, and he showed that Clarkson's hypothesis, for the first time, he showed without question that Clarkson's timing hypothesis was transferable from a primate to humans. So we know now that there's a fixed window that we have to understand, and we have to understand it if we're going to understand if vascular dementia is involved in, estrogen's involved in vascular dementia. Victor Henderson proposed that there was a similar timing hypothesis for, he proposed it for Alzheimer's. One might say he was probably talking about dementia, but in any case, so it was proposed. And now what I'm going to tell you at the end of, or through this lecture, is that we propose now that there's a window for estrogen protection against dementia, but it's maybe limited to the prevention of vascular dementia, not against Alzheimer's. And here I have, and I'm sorry I don't have the attribution, I simply couldn't find it on my PubMed, but it's now a quite important and well-known paper, which is the one that Andrea referred to in his lecture. And this is a retrospective study in the Scandinavian countries, and it's really well done. I think everybody should read it. It's because it shows you how you do these things. And what I wanted to talk to you about today is their findings. Now, they took a group of men and women, and they matched up the cause of death with their registry of taking medications. That's how they did this study. And so this is big data. This takes a certain kind of epidemiology. And I put in the red box what's important. You see that when you get to take hormones for 5 years or more, you begin to see down at the bottom, you begin to see that now you have evidence where the confidence limits do not cross 1. So that means these are acceptable relative risks. Up above, when you took less than 5 years of hormones, you may have had a preventative effect, you may not. The numbers are not all that different. The protective effect, less than 5 years, but they all cross 1. So they're not statistically significant. But when you take it for 5 years or more, you get statistically significant protection against, again, they say vascular dementia and Alzheimer's. Again, they're taking patient care records, most of which have never had imaging and certainly don't have, they said they had a lot of postmortem biopsies, but I would say for the moment we should think we're talking about dementia, and taking hormones for 5 years or more is what seems to have done it, and in other parts of the paper they show that this depends on when you start, around the time of menopause, not when you're 70 or 75 years of age. So, taking all of this into account, that we have dementia, we don't know what it's caused by. We have cardiovascular protection. We know estrogen confers it. Can we start to weave this into our clinical practices, into our patients, to decide when and who and how long to treat? So I've made a cartoon for you of how I see the way that women go through the stages of life. I've curtailed it to the perimenopause, so you have the age at the bottom, and you can see in the center the milestone, the menopause, and you can see that I've included underlying subclinical disease up until the time of menopause, because women do come into menopause with subclinical disease, and then the growth of subclinical disease until something like 15 years later, they have clinical disease, and you see that on your right. I called it brain dystrophies because, again, I'm not sure what it means. So you can see the timetable of these things happening. And if we were to then put that cartoon against a plan for treatment that could be useful, you can see how, just following it along, don't be confused by the red stuff at the bottom, except to see that education, early intervention, prevention, and then comes treatment. And treatment comes before clinical disease. You see that little dotted line? Treatment starts earlier than you have clinical disease. We are not to wait until we have somebody who has dementia. We are not to wait until we have somebody who has a cardiovascular event. And so you can see this as a proposed, at least a place to start talking about how to employ what we've talked about. So in summary, what I've told you is that dementia is common and getting more common. That although it has many causes, vascular dementia and Alzheimer type dementia are the most common, and they may coexist. That vascular dementia is due to vascular disease, while Alzheimer's is a neurodegenerative condition. They are different diseases that have a common outcome. We are looking at the outcome. It's kind of like we're Indian scouts going around looking and seeing what the animal droppings are telling us about who was there before. And that what we have to do is to do imaging studies during life to make diagnoses if we really want to get solid data. But in the absence of solid data, we have to look at what we know. We know that there's a prevalence of dementia in women over men. We know that estrogen prevents atheroma and ultimately, we assume, atherosclerosis. And there are now 1 or 2 papers from one from the WHI, which has now gone on long enough to actually see a difference in vascular lesions, clinical events. Estrogen or menopausal hormone treatment does not affect minimal cognitive dysfunction, and we and others, the KEEPS trial showed it, and others have shown this, and minimal cognitive dysfunction is what is associated with Alzheimer's. Another mark against estrogen improving Alzheimer's. So we have to think about, and when one compares the ability to protect against vascular disease or vascular dementia and not against Alzheimer's dementia, the papers that are published appear to fit with that general idea that what we're doing is treating the cause of the dementia. We're just mistaking the cause of the dementia because we're looking at the outcome. Estrogen treatment with women with dementia clearly is too late. If somebody has dementia, it's too late. So let's think about what we can do. We can think about preventing by using vascular cardioprotection the same way we do now. Think about it for menopausal hormone treatment. And we should actually do that. We should make studies to do this. And since we know from particularly the studies of Howard Hodis, when he did the ELITE trial, that starting the hormone treatment after 6 years from the menopause does not protect, at least in a discernible way. We have to think about starting before 6 years, 10 years, whatever it is, doesn't really matter. The idea is to understand that menopausal hormone treatment should be given to menopausal women, not postmenopausal women 10 years down the line. So I would urge you not to try to swallow this whole. There's a lot of information here, but really think about it and think about what it is that we are doing and perhaps what we are not doing for women. And if you know that you cannot see a difference with less than 5 years in the Cache County trial, and if you know that you start at 6 years or earlier, according to Howard Hodis' outcomes with ELITE trial, if you're dealing with vascular disease, then you will ultimately come to the conclusion that you have to start hormone replacement early, that you cannot individualize it for hot flushes, because you don't even know what causes dementia. You have to decide you're doing it as preventative treatment, and you better do it for a long, long time. Thank you very much.